Good day, everyone. I am Mr. Molale from Ikailelo Secondary School. I am a departmental head and also a senior marker for Life Sciences Paper 2. So I am here to just assist you with the key points for the June preparation examination. And it should be known that the mark allocation that you are going to see on the screen, or the marks that I'm going to say, it's for the Free State province. So those marks are only for the Free State. So let us not waste time, since we only have 60 minutes to cover everything. So basic technical points, we will finish with them. Ne? I just want to start with the important stuff. The first topic that you must know will be on the paper. It's DNA, the code of life. Please just ignore the marks there. Ne? It must be 60 marks. So everything DNA, the code of life, it will be 60 marks from question 1 up to the last question. Now, what's important for you, ladies and gentlemen? Let us know the functions of DNA. And remember, the functions are 2, for the making up of genes and also to carry what? The hereditary information. And then also the DNA, we know that it codes for protein synthesis. It contains the coded information for protein synthesis. Also, let us know the role of DNA. What role does DNA play during the process of transcription? So the role is that during transcription, we know that one strand of the DNA molecule acts as a template to form complementary mRNA. And the mRNA is the one that will carry the role of mRNA. It is to carry that coded information from DNA and transfers it and takes it to the what? To the ribosomes. So we must know the role of DNA in protein synthesis. We must know the role of mRNA in protein synthesis, and also the role of tRNA in protein synthesis. Moving forward, we look at the difference between DNA and RNA. Now, the mistake that learners make is this one. The question would say, give 4 structural differences of DNA and RNA. And then we would find learners giving us the function. If it says structural differences, it means we only want the differences in terms of what? The structure. The structure of the 2. I don't want to talk about paper 1. You know what you did, okay? Structural difference. Please don't make the same mistake should we have a question on the differences. If they ask for functional differences, then you give the functions of each. That will be the functional differences. So when we look at the differences of DNA and RNA, now, your differences should be in a form of a sentence. What we don't want to see in your answer sheet, we don't want to see deoxyribose, ribose. It must be the sugar is deoxyribose. In DNA, the sugar is ribose, not deoxyribose, ribose. Do you understand? Also, the nitrogenous base in DNA is thymine, whereas in RNA, the nitrogenous base is uracil. Not thymine, uracil. What's thymine? What's uracil? It's nitrogenous what? Base. Ne? So these are the what? These are the structural differences. And a difference in monomers. We all know what are monomers, right? Monomers are the building blocks of what? Of nucleic acid, of nucleic acids. Okay? What are monomers of nucleic acids? It's nucleotides. So you must also be able to differentiate between a nucleotide of a DNA and the nucleotide of an RNA. So we know that the nucleotide consists of 3 parts, right? For DNA, we must mention it consists of the phosphate, right? Then the sugar is deoxyribose. Then the nitrogenous bases, you mention all 4 of them, which is adenine, thymine, guanine, and cytosine. Then, how are they paired? How do they bond? We know that the phosphate bonds with what? Oh, sorry. Is it visible for everyone? The phosphate bonds with the what? With the sugar. But this one is the DNA what? Nucleotide. So the phosphate group will bond with the deoxyribose sugar. Please don't write DS. Deoxyribose sugar. Then the deoxyribose sugar will bond with the what? With the nitrogenous what? Base. I'm also giving you a heads up if maybe they say you must describe the structure of DNA. When we describe the structure of the DNA, we must mention that the DNA is a double helix molecule. The natural shape, right? Then the DNA is made up of monomers called nucleotides. The phosphate connected to the deoxyribose sugar. The deoxyribose sugar connected to the what? To the nitrogenous base. Then we know that in the DNA molecule, there is the pairing of the what? Of the nitrogenous base. Again, where we must know that adenine pairs with what? Thymine. And then guanine pairs with cytosine. Then that is the structure of what? You are describing the structure, then in between the nitrogenous bases, the bond there is what? Hydrogen bonds or weak hydrogen bonds. If you know how to describe the structure of DNA, then you will also know how to describe the structure of RNA. We know that RNA single strand. The nucleotide phosphate, it bonds with the what? With the ribose sugar. The ribose sugar will bond with the what? With the nitrogenous base. What are those nitrogenous bases? Adenine, uracil, guanine, and cytosine. And please, let us stop saying that in RNA and when we answer the questions on mRNA and tRNA, let us stop saying adenine pairs with uracil. Adenine is complementary to uracil. They don't pair because RNA is single-stranded. RNA is single-stranded. Are we good with that, ladies and gentlemen? Another thing that we must know, since in DNA we know that we have the pairing of the nitrogenous base, again, we know that adenine is equal to what? To thymine. And guanine is equal to cytosine. Now, if we have 20% of adenine, they can say you must either find the percentage of guanine or cytosine. And what does that mean? If you have the percentage of adenine, you already have the percentage of thymine. So if adenine is 20, meaning thymine is also 20%. Then you have 20% plus 20%, which is equal to 40%. Then you say the 100 minus 40%, which will give you bukai 60%. You don't end there. This 60% must be shared by what? By guanine and cytosine. Then you divide that 60% by 2, which is going to be 30%. Then 30 will be for guanine and 30 will be for cytosine. When we answer multiple choice questions, especially for paper 1, please do note that before you get to the answer, some of these multiple choice questions, you have to work them out before you get to the answer. You are not going to play lotto whereby you just choose A, B, or something. You must work them out before you get to the answer. Ne? And then others, the question might be, what is the ratio to guanine and adenine? The ratio for guanine to adenine. It cannot be 30:20. We have to simplify. The ratio will be 3:2. Is it clear, ladies and gentlemen? And then also, if the DNA molecule has 200 nitrogenous bases, then from the 200 nitrogenous bases, 20 of them are guanine. How many nitrogenous bases are there for adenine? You also have to do that. If they're giving you that guanine is 20, you will know that adenine is also 20. You have 40. You just say 200 minus 40, which is equal to 160. You divide that 160 by 2, then it will give you the number of the nitrogenous base that is requested. And also, if the DNA molecule has 20 nitrogenous bases, how many sugar molecules are there? How many deoxyribose sugars will be in that molecule? There are 20 nitrogenous bases. How many sugar molecules will be there? There will still be 20. But you will know how to answer that if you know the monomers of nucleic acids. Thank you. Then we move to the process of protein synthesis. Protein synthesis, we must know that it has 2 stages. The stage of transcription and the stage of translation. Sorry, we are now doing DNA replication, not, sorry, ne? So DNA replication, I'm in a hurry, sorry for that. DNA replication first before protein synthesis. So what we must know regarding DNA replication, we must know when in the cycle it takes place. When? It is during interphase, right? And then where in a cell it takes place? It takes place in the nucleus. It takes place in the nucleus. And then how? How? 6 marks. The process of DNA replication. Please, no names of enzyme is required. You don't have to mention the enzymes. You just mention the word, the process, and also let us know the significance. Why is it important that the DNA should make an identical copy of itself? We know that DNA replication is when the DNA makes an identical copy of itself, right? Now they can say, describe the process where the DNA makes an identical copy of itself. And then someone will be like, oh, now I know DNA replication. Now what are they asking? So you must also know how to define those processes. It occurs in the what? In the nucleus. From one DNA molecule, if you are given a diagram, from one DNA molecule, how many molecules will be present at the end? 2, and they must be identical to the original one. That is DNA replication. Now, how? How does the process occur? The DNA double helix or the double helix DNA unwinds. If you just write the double molecule unwinds, no mark. Or if you just write the DNA unwinds, no mark will be awarded. It must be the DNA double helix. It must do what? Unwinds. Okay? Then the mole, we know that the weak hydrogen bonds do what? They break, and then the molecule unzips to form two separate strands, right? Then from the two separate strands, both strands, they do what? They serve as a what? As a template to build a what? A complementary DNA. To build a complementary DNA using what? Free DNA nucleotides from the nucleoplasm, resulting in two identical DNA molecules, not two identical DNA strands. It must be two identical DNA molecules. Is it clear, ladies and gentlemen? Yes. So, that is DNA replication. The significance, don't forget the significance, again. It doubles the genetic material, again, so that it can be shared by the what? By the new cells arising from cell division. So let us know the importance of DNA replication. Moving forward to protein synthesis, we have two stages there, which is transcription and translation. Now, transcription occurs in the what? In the nucleus. Now, what is transcription? Transcription is the synthesis of mRNA from DNA. We are forming what? mRNA from DNA. How? The DNA double helix unwinds. Again, the two strands unzips because their weak hydrogen bonds have broken. Again, then how many strands now act as a template? One. Just one strand acts as a template to form what? mRNA. Using free RNA nucleotides from the nucleoplasm. Then mRNA is complementary to DNA. mRNA now has the coded message for protein synthesis. Please, let's know the process. Translation. Can any one of you just tell me where is the site of translation? It's in the ribosomes, not the cytoplasm. It's at the ribosome. This process occurs at the ribosome. It is the site for translation. Now, translation, please, ladies and gentlemen. Each tRNA, it carries what? Oh. Each tRNA carries what? Carries a specific amino acid. That specific must be there. It carries a specific amino acid. And then when the anticodon on tRNA, not when the codon matches with the, not when the codon matches with the codon. It must be when the anticodon on tRNA. Anticodon and tRNA must be on that sentence. Matches with the what? With the codon on mRNA. Then what does tRNA do? It brings what? The required amino acid where? To the ribosome. Don't just say tRNA brings the required amino acid. It must be to the ribosome. Right? And then we know that our amino acids are attached to each other by peptide bonds to form the required what? Protein. To form the required protein. Not just to form a protein, to form the required protein. Now, the roles of tRNA in translation or during protein synthesis, you take them from this process. Kiena, the first one. tRNA, what does it do? It's the function. It brings what? The required amino acid to the ribosome. What does tRNA also do? The anticodon on the tRNA matches with the codon on mRNA. tRNA also carries a specific amino acid. So we have two RNAs, mRNA and tRNA. Again, mRNA is formed during transcription. We use tRNA during translation. Note the structural differences of the two. mRNA, it's long, it has unlimited what? Unlimited nitrogenous bases or unlimited what? Codons. Whereas tRNA, it's like a hairpin, right? And then it only has how many nitrogenous bases? 3 nitrogenous bases or 1 codon. Or 1 codon. Then, this diagram, ladies and gentlemen, familiarize yourself with, know how to label those parts. The first one, when we look at it, this is what? The DNA, right? This one, it's a process because they are showing us it's from this to this. Then the process will be what? Transcription. What is this molecule? It's single-stranded. The molecule is what? mRNA. The process occurs in what? This is the nucleus. Okay? So what is this structure? The double nuclear membrane. You must know we have that thing that is dotted inside the nucleus, which is the nucleolus. Okay? We know the fluid inside the nucleus is the nucleoplasm. I know some of you are always saying using free RNA or DNA nucleotides from the nucleoplasm. You don't even know what is the nucleoplasm. The nucleoplasm is the what? It is the fluid in the nucleus. Good. Also, we know this will be what? This is our mRNA. It attaches itself on the what? On the organelle ribosome. And then we have tRNA. It carries what? Amino acid. This is the tRNA. The 3 nitrogenous base on tRNA is anticodon. The 3 nitrogenous base on mRNA is codon. Let's know that, ladies and gentlemen. You see, you will be given a diagram like this maybe, and then they say, what is that molecule? DNA, right? What is that one? mRNA. Then this is the double nuclear membrane ribosome. It's 1, 2, 3. It's what? It's a codon. It's mRNA because it attaches on the what? On the ribosome. This is what? It's amino acid. It's on top. It is carried by what? By tRNA. And then this molecule from here to here, it's what? It's tRNA. Do you see the difference, ladies and gentlemen? Yes. And then, on this process of protein synthesis, mutations, ladies and gentlemen, gene mutation, they change in the sequence of the nitrogenous base on DNA or mRNA or tRNA. Again, now, we know that you will be given a base triplet, and then they will say that base triplet, A or T, is replaced by another nitrogenous base, right? So you have to be able to explain how will that mutation have an effect on the what? On the protein that will be produced. On the protein that will be produced. Now, also, from the table, if the table here, you see, here is our table, ladies and gentlemen. We are given DNA what? DNA-based triplet, and then we are given amino acids. We can use amino acids to find what? To find anticodons. Right? We are given DNA and amino acid, but they request us to find what? The DNA-based triplet of leucine. We are given DNA-based triplet, amino acid, but they want us to find what? The anticodon on tRNA for leucine. What must we do? The table has given us what? It has given us the base triplet of DNA. Okay? So for us to get to anticodon, we first have to find the what? The codon. The mistake that you do, if you are given DNA and then they say provide, let's make an example, ladies and gentlemen. So we have leucine. What is the DNA? It's GAC, okay? Which is leucine. So we have GAC as DNA. Then they request us to find what? The anticodon. So if this is DNA, we must find the what? mRNA, which is going to be what? CUG. After that, you find the what? The anticodon, which is tRNA, which is going to be GAC. We don't want to see this. I agree, they just requested you to write the anticodon. I agree. We don't want to see this in your answer sheet. We just want to see the answer. The anticodon, not this. Is it clear? Good. And then for the mutations, this is what I did. I hope it will assist you, ladies and gentlemen. Now, you will be given, you can see that I just labeled it as how to answer gene mutation. You can see here we have nitrogenous bases. Ne? We have before and we have what? After. Right? So our DNA-based triplet was TC gene. And then the G was replaced by C. Do you see that? So now, how do we answer this? We do the before, everything from DNA up until amino acid. Then we do the after for both. And then you put this in a form of a paragraph or explanation, whereby you will say the DNA-based triplet, which one? TC gene, changes to TCC. It changes to TCC. Then second point will be what? mRNA codon AGC changes to AG gene. Now, when you get to this tRNA does not change. But what does tRNA do? It brings the required amino acid way to the ribosome. So it means what? The anticodon on tRNA will bring what? It will bring valine, the one after, instead of what? Instead of proline, leading to the formation of a different ink protein. Is it clear, ladies and gentlemen? The DNA-based triplet, TCG, changes to TCC. The mRNA codon changes AGC to AGG. Then tRNA, think of the word, the function. The anticodon tRNA UCC, because we want the one that is going to be broad. It's not proline, it's going to be what? Valine. So the anticodon on tRNA will bring valine, which is on the after, instead of proline, leading to the formation of a different protein. Then if you get to the amino acid, and then the amino acid is still proline instead of valine, it means you must state that tRNA will bring the same what? Amino acid, which is proline, and then leading to the formation of the same protein because the amino acids are the same. Is it clear, ladies and gentlemen? Thank you. Now we move to DNA profiling. Mahala Max. You will be given a diagram like this, and then there are DNA profile bars, right? Now, please use a ruler. Don't say I will just look at it with my eyes and I will count those DNA profile bars or bands. Use a ruler to tick them. Name. So we have the baby. When we look at the baby, there's the baby. Matches with the what? These are the ones that matches with the what? With the mother. These ones, they matches with the what? With the mother. And then these ones in green, they match with what? With the father. And then we count. We have male 1, male 2. We have male 1 and male 2. Who is the father between male 1 and male 2? It's male 2, right? Why is he the father? Okay, all right. Then you must say all the DNA profile bars of male 2 matches with all the DNA profile bars of the baby. I'm coming to also how you can answer it. And then also you can say, and then you add by saying the remaining DNA profile bands matches with those of who? Of the mother. Again, what you can say is that 1, 2, 3, 4. You can mention their numbers by saying 4 DNA profile bars of male 2 matches with 4 DNA profile bars of the baby. Then the remaining, how many are for the mother? 3. There are 4. Yes, then the remaining 4 is for who? It's for the mother. The remaining 4 is for the mother. So, as long as you are able to answer that question, is it clear for that one? And please, not just DNA profile. It must be DNA profile bars or DNA profile bands. Only the 2. Only the 2. And then also we must know the use, ne? The use. Please stop for this one. Stop saying to solve crimes. We are not solving. We are investigating crimes. We identify organisms from their remains. Again, we don't just say for organ transplant. Don't just say for organ transplant. To establish matching tissues for organ transplant. It's clear, ne? All right, so here is just to show you the matching there. Then we will be done. We are actually done with what? With DNA. Then again, now we move to the second topic, which is meiosis. Ne? Meiosis, please ignore that 21. The mark allocation should be 30. 30 marks. Ne? And remember, this is for the Free State. Free State. Ne? Then meiosis, the importance. It produces what? Haploid gametes. It halves the number of chromosomes. It also introduces what genetic variation. How does meiosis introduce genetic variation? By 2 processes, which is crossing over and random arrangement of chromosomes. Crossing over, ladies and gentlemen, it occurs during when? Prophase 1. You must hold in this point. It occurs during prophase 1. It's a mark. Chromosomes are arranged as homologous. Then, non-sister chromatids, not just chromatids. It must be non-sister chromatids overlap, or you can say adjacent chromatids overlap at the point called what? The chiasma or the chiasmata. And then to exchange the genetic material, resulting in a new combination of genetic material from both parents. This is crossing over, and then random arrangement of chromosomes at the equator. We know it occurs during metaphase 1 and metaphase 2, right? During metaphase 1, chromosomes are arranged in homologous pairs at the equator. Then during metaphase 2, just single chromosomes are arranged singly along the equator, right? Yes. And also, what you must know, the chromosomes. On the chromosomes, we have genes. What is a gene? A gene is a segment or a piece or a portion of what? Of DNA on a chromosome. So let us know that. And also the number of chromosomes in humans, we know somatic cells, they have 46, while haploid, while gametes have haploid number, which is 23. Then chromosomes are single-stranded and they are held together by centromere, chromatids, centromere there. Then, for this one, this is a chromosome. How do we know it's a chromosome? It has a centromere, then this centromere attaches the two what? Sister chromatids. Now, this one, this is during anaphase, ne? During anaphase, if they are like this, we call them what? Chromatids. During anaphase, man, 2. You only call them chromatids during anaphase 2. But when we get to telophase 2, we do not call them chromatids. We call them daughter chromosomes or unreplicated chromosomes. So if they are like this, this thing, both of them, it's an unreplicated chromosome, it is an unreplicated chromosome. Why do we say that? Both of them, they have what? A centromere. A chromatid does not have what? It does not have a centromere. A chromatid, it is only a chromosome that has a centromere. So even if you see them like this and then you see that there's centromere, you must know that it is a chromosome that is unreplicated or a daughter chromosome. So if they ask you if they are homologous, will that be yes or no? Are these homologous chromosomes? Are these homologous chromosomes? Yes. Even though they are single-stranded. It's a chromosome. It's a chromosome. It has what? It has a centromere. Why are they homologous? They have the same size. They have the same shape. They have the same position of the centromere. That is why we say they are homologous chromosomes. And then the difference between mitosis and meiosis. Please go and study this. We know that meiosis involves 2 cell division, while meiosis involves 1 cell division. Go and study those differences. The difference between mitosis and meiosis. Then you must be able to differentiate between haploid and diploid. We know haploid cells are gametes, while diploid cells are somatic cells. So be able to differentiate between the two, haploid and diploid. Then homologous chromosomes, you are used to them as like this, right? Remember, they can also be single-stranded, both of them. As long as they are single-stranded and they have a centromere, both of them, we regard them as chromosomes. So we say they have the same what? Shape, not the same structure, not the same morphology. They have the same size or length. Don't say same size, number one. Number two, same length. It's one mark. They also have the same position of the genes at the same locus. We know that, or loci, right? Then the genes codes for the same what? Characteristics, and they have the same position of the centromere. Behaviors of chromosomes during different phases. You must be able to identify those phases and be able to explain why are you saying is that particular phase. Why do we say is that particular phase? So we have what happens during prophase, metaphase 1, anaphase 1, all of them. Also, the diagrams, let us be able to label. Let us know how to label the centromere, the spindle fibers, also the functions of the labels. Also the functions of the labels. And if it is metaphase 1, we do not say homologous chromosomes present and line at the equator. We must say homologous chromosomes arranged in pairs on the equator. Not homologous pairs arranged on the equator. Please. Then this is just questions on different phases, how to identify name. Then genetics, which is the last part, 60 marks. 60 marks as well. Ne? And remember, it's for the Free State, 60 marks. Not 72, ne? 60 marks. Then biological terms. Know your biological terms. The difference between a chromatin and a chromosome, a gene and an allele, and when we get to alleles, please do not forget we also have multiple alleles. Ne? Yes. Phenotype and genotype, dominant and recessive, homozygous and heterozygous. So they can just use these words instead of this one. You must know if they say it is pure breeding, you must know it's homozygous. Ne? Same with heterozygous. Monohybrid cross and dihybrid cross. If you don't know the format of monohybrid cross, you are going to have a problem on Monday. So these are just the definition of those hormones. Then you must know the relationship between DNA, a gene, and a genome. The difference. So a gene is a portion of DNA on a chromosome, which codes for particular characteristics. Whereas DNA, we know that it is the one that carries the hereditary information. Then genome is the what? Is the mapping of all the genes. The mapping of all the genes. So you must know the full definition of what a genome is. Then this one is a karyotype. Know what is a karyotype? A diagram which shows the arrangement of chromosomes according to their number and the shape in the nucleus of a somatic cell. So know what is a karyotype. The origin of the chromosomes. If we have chromosome 23, we know that one chromosome is from the mother and one chromosome is from the father. Then this is a genome. Ne? The mapping of the exact position of all the genes in the chromosomes of a human. It's a genome. And then do not forget also about multiple what? Multiple alleles. More than 2 alternative forms of a gene at the same what? Locus. Example, they say what? Blood groups. We know that blood groups are controlled by 3 alleles. And these 3 alleles, they can give us how many blood groups? They can give us 4 blood groups. Which is blood group A, blood group B, blood group AB, and blood group O. Now, you must be able to explain the types of dominance using blood groups. The type of dominance using blood groups. For example, which type of dominance do we have for blood group AB? It's co-dominance. Again, why do we say it's co-dominance? Because both alleles are expressed. 0 mark. 0 mark. It's blood group AB. Why do we say it's co-dominance? It's because allele A of a gene and allele B of a gene are equally dominant. It must be like that. Don't just say both alleles. It's AB. It has 2 alleles. Allele A and allele B. If we have a flower that is red and we breed it with a flower that is white, and then the flower is red and white, are we going to say it's called dominance because both are equal? No. The white allele of a gene and the red allele of a gene are equally dominant, such that both the alleles of a gene, red and white, are expressed in the phenotype in a heterozygous condition. That application is very important. The type of dominance for blood group A, it's if we have heterozygous. If we have heterozygous, the type of dominance is what here? It's complete dominance. Why is it complete dominance? This allele, you say allele A is dominant. You cannot say one allele is dominant, the other is recessive. Which one is dominant? Which one is recessive? State that in your answer. You understand what I'm saying? Also, we also have the following, ladies and gentlemen. We also have Mendel's law of dominance. Okay? Mendel's law of dominance. If we have this allele with this one, this is in a heterozygous condition. We cannot say one allele is dominant while the other is recessive. State that the allele tallness is what? Dominant. While the allele short is what? Recessive. Then tall, which is dominant, what does it do? It masks the shortness, which is recessive. That application, Mendel's law of segregation, it says it consists of what? 2 factors, right? And then they separate again, so that each gamete must have the 2 again. So if you explain that using Mendel's law of segregation, you must state that there are 2 factors. What are those 2 factors? These 2 allele. Allele this one and allele this one in the bracket. To show what you are talking about. What's going to happen? These 2 alleles are going to do what you mentioned. They separate. When they separate, what happens? It means a gamete can have either this allele or it can have either this allele. Then application. Application, very important. Is it clear, ladies and gentlemen? Yeah, unfortunately, our time is about to end, but we have an idea, right? So, in terms of karyotype, I spoke about the origin of chromosomes. Ne? At position number 3, we can see that one allele is from the mother and one allele is from the father. Okay. But when we get to position number 23, look at position number 23. This is XY. Are we still going to say one from the mother, one from the father? We must say the X chromosome is from the mother. The Y chromosome is from where? The father. Sex determination. Right? Yes. And then types of dominance, I have already explained, right? You will see here, for complete dominance, red allele is dominant and white is what? Recessive. Not just one is dominant, one is recessive. Application. Ne? And then those are the types of dominance. Ladies and gentlemen, the cross, very important, monohybrid cross. The template, just like that. And then dihybrid crosses, we know they want the gametes, the phenotype, the genotypes. Okay? Then, the phenotype, if we have tall plant which has green flowers, you cannot just say tall and green. It must be a tall plant with green flowers, not just tall and green. Is it clear? When you are asked to give an allele is only one letter. Allele is only one letter. Different laws, I mentioned them. Application, right? Application, all three of them. Also multiple alleles, we spoke about it, how sex is determined. And then on sex determination or also sex-linked disorders, we must also know why males are more affected than what? Than females. Why are more males affected than females? Interpretation of pedigree diagrams. You know, for a pedigree diagram, ladies and gentlemen, what you must just identify there, you must know whether the condition is caused by a recessive allele or whether it's caused by a dominant allele. Then you will be able to complete the genotypes of all the individuals on those pedigree diagrams. The generations, how many generations are there? The relationship. Why is this child having hemophilia? Why is this child having an extra finger? You must be able to state the parents are like this. One parent provided this allele. The other parent provided this allele, leading to a child being like that. Very important. So those are just the steps to follow and different questions. Sex-linked disorder, why are more males affected? Ajir, it's the same as why females have a smaller chance, guys. It's the same. And then mutations, we have done that. We are good, right? Stem cell research, know the definition of stem cell and also the sources as well as the uses. Cloning, ladies and gentlemen, as well. And then another thing, why are blood groups not conclusive? When you do or when you go for a paternity test, why are blood groups not conclusive? Very important. It's because blood groups can tell who is not the father, but they cannot tell who is the actual father because many men share the same blood group. Therefore, DNA profiling is the more conclusive since the DNA consists what? The genetic or hereditary information.